All right, we can get started as people are trickling in. Um, I just want to welcome everyone today. I'm Leanna Orlando. I'm the VP of Research at CureDuchenne, and today we're going to hear from Dyne Therapeutics, who has a exon skipping platform in a program for skipping exon 51 and some initial clinical data to share with us today. But before we get started, I wanted to handle some housekeeping information. We will have a Q&A at the end of the webinar, so anytime during the presentation, you can enter a question using the Q&A button at the bottom of the screen. If you're not seeing the Q&A button at the bottom of the screen right now, just move your mouse down to the bottom and it should appear. And so you can enter your questions through there, and we're going to do our best to get to all of them. I will note that anything about a person's specific, clinical case or mutation is really more private in nature. So if you have those kinds of questions, we will help you follow up with Dyne and the CureDuchenne care team afterwards. But otherwise, feel free to enter any other types of questions. This webinar is being recorded, and after today, it will be available on the CureDuchenne website in case you'd like to share it with anyone else you know or to listen again. So with that, I'd like to invite Ash to the screen to meet our first speaker, Ash Dugar, who is the Chief Medical Affairs Officer at Dyne.
Great, thank you, Leanna. So as Lana said, my name is Ash Dugar, and I'm the Chief Medical Affairs Officer here at Dyne Therapeutics. First, I'd like to thank Deborah and Paul Miller and the entire CureDuchenne organization for this opportunity to present what we believe is an exciting update on our Duchenne program to the community. Before I begin, I'd like to introduce you to our new head of patient advocacy, Catherine Beaverson. She's on the screen here. As many of you know, Catherine has been part of the neuromuscular advocacy community for many years and brings tremendous experience and passion to the role. Please know that Molly White, who was our head of advocacy, isn't going too far. She's simply taking on a new role here at Dyne. So I'm really excited for this addition to our team with Catherine's arrival, while continuing to be able to work closely with Molly in her new role. And I have to thank Molly for building Dyne's key pillar, our delivering for patients core value, and connecting Dyne to this incredible community that we try to serve. And Catherine will be back at the end when we do Q&A. Thank you.
All right, thanks so much, Ash.
Okay. Next slide, please. So I'd like to remind everyone that we will be making forward-looking statements today, and you can refer to our website for more information. Next slide, please. At Dyne, it all starts with our mission to deliver life-transforming therapies for people living with serious muscle diseases. Our mission is woven deeply into our core values, and it inspires our employees, our dynamos, each and every day. Next slide, please. So the intentional design of the FORCE platform, which I presented to this community a few times, seems to be translating in the clinic from a safety as well as an exon skipping and dystrophin production perspective. And I'm pleased to show those results a little bit later in the presentation. By way of reminder, the FORCE platform drives our efforts to develop and deliver muscle-targeted therapeutics. It is expressly designed to address one major issue in muscle diseases, which is efficiently and reliably delivering antisense oligonucleotides to skeletal muscle, including diaphragm, cardiac, and smooth muscle, and target the genetic basis of disease. It consists of three components: the Fab, or the antigen binding fragment, or the antibody fragment, which is the top portion of this Y-shaped figure on the left-hand side of the slide. The Y-shaped figure is a full monoclonal antibody. So what we've done is taken the top portion of this monoclonal antibody, again, the Fab, and designed it to bind to something called the transferrin receptor 1, or TFR1. The TFR1 receptor is highly expressed on the surface of muscle cells, such as skeletal muscle and cardiac muscle. The second component is the linker, which you see on the bottom left. We chose this clinically validated linker because of its demonstrated safety and efficacy in third-party approved products. And this linker enables precise connection of the Fab to the antisense, and in this case, an exon skipping PMO. And so that third component, in this particular case, is an exon skipping PMO, which we rationally selected to target the genetic basis of Duchenne. This is where we leverage the modularity of the FORCE platform. What I mean by that is for nuclear targeting diseases such as Duchenne, we choose antisense oligo or ASO payloads. And in this case, we chose an exon skipping phosphorodiamidate morpholino oligomer or PMO that targets the specific mutation in the DMD gene. And Dyne 251 is our investigational medicine that uses an exon skipping PMO designed to enable skipping of exon 51 to make a shortened functional dystrophin. Our FORCE platform is designed with the goal of addressing current limitations of delivering antisense oligos to muscle tissue, to improve the delivery of the PMO to cardiac tissue and skeletal muscle, including diaphragm, and to allow for increased time between doses. Next slide, please. Now, the mechanism of FORCE is different from other approaches. We are essentially using the cell's natural biology to deliver our investigational medicine to muscle cells. As I mentioned, our Fab binds tightly to the TFR1 receptor, and TFR1 is highly expressed across the surface of muscle cells, including cardiac, skeletal, and smooth muscles. Now, the Fab acts as a kind of key that unlocks the TFR1 receptor and allows our PMO to enter those muscle cells to target the genetic basis of disease at the level of the nucleus. So the FORCE platform harnesses this natural mechanism of the TFR1 receptor mediated delivery to transport the PMO across the cell membrane, as you can see in the schematic. And this is differentiating because we are not using membrane destabilizing agents to deliver the PMO into the cell or to leave the endosome, which you see in the middle part of the slide. And so we are able to actually have a mechanism that allows us for a potentially wide therapeutic index, resulting in less frequent dosing and chronic administration. Next slide, please. So today, Dyne is well on its way towards achieving the potential of FORCE and delivering for patients. We've delivered exciting clinical proof of concept data for our Duchenne program, and I'm pleased to share our initial data readout. Next slide, please. So prior to starting our Dyne 251 clinical development program, we proactively obtained expert feedback. We engaged key opinion leaders, or KOLs, from various disciplines, including pediatric and adult neurology, cardiology, physical medicine and rehabilitation, pulmonology, and physical therapy. And importantly, we gathered this feedback from across the globe. KOLs, both in the US and outside of the US, with experience running early and late stage clinical trials, have provided advice on our overall clinical development program, including our DELIVER study, such as advice on our clinical trial design, the patient population that we're studying in this phase 1/2 study, biomarker and functional endpoints to include, and key safety considerations. Importantly, they have also provided key learnings from numerous long-term natural history studies to help inform our trial design and eventually to contextualize our results. It is clear that the KOLs believe dystrophin continues to be a key endpoint for this condition. We've also received advice directly from patients and caregivers, as well as global advocacy leaders such as CureDuchenne and PPMD, and also from the Duchenne Community Advisory Board, comprising of advocacy leaders from several countries around the globe. They've provided their insights based on their living with and being the voice of the Duchenne community, as well as their experience with past and current clinical trials. This input has been absolutely critical in ensuring a patient-centric design for DELIVER and has included, but is not limited to, key considerations when families are choosing a clinical trial because there are so many choices. Review of and input to our informed consent form, feedback on biopsies and functional outcomes, identification of the most burdensome or difficult parts of the trial, and how to ensure patients and families are supported throughout the process. Next slide, please. So before I go into detail on the data, I want to provide a brief review of the DELIVER study design. It is a phase 1/2 study, global clinical trial that we have designed to be potentially registrational. The trial is enrolling ambulant and non-ambulant males with Duchenne who are ages 4 to 16 and have mutations amenable to exon 51 skipping therapy. Patients who have been previously treated with an exon skipping therapy can enroll and deliver after a 12-week washout period from their current exon skipping treatment. The primary endpoints are safety, tolerability, and change from baseline in dystrophin levels as measured by a technique called Western blot. Secondary endpoints include measures of muscle function, like NSAA and time function tests and exon skipping. We also have wearables to allow collection of data such as stride velocity. Next slide, please. So the DELIVER trial consisted of 4 parts. There's a 6-week screening period, a 24-week multiple ascending dose or MAD randomized placebo-controlled period, a 24-week open label extension, and a 96-week long-term extension. Now, in the MAD placebo-controlled portion of the DELIVER trial, participants are randomized to receive Dyne 251 or placebo every 4 weeks. And then all participants receive Dyne 251 every 4 weeks after the MAD portion. Again, we have a number of outcomes measures to evaluate function and quality of life. And for details on the outcomes, as well as the comprehensive set of inclusion and exclusion criteria, you can visit our ct.gov page for more information, and that number is listed at the bottom of the slide. Again, we have a number of sites in the US and outside of the US. Next slide, please. Now I'll review the initial data results. We're very pleased with the favorable safety profile of Dyne 251 that we have observed and DELIVER as of the data cutoff date. Now, we believe this is differentiating from others in the field and speaks to the importance of each component of the FORCE platform, as I mentioned earlier. The Fab, which is just that portion of the monoclonal antibody, the clinically validated linker, and the choice of the exon skipping PMO. Overall, the safety profile of Dyne 251 has been favorable through the data cutoff date. The safety data is based on 37 participants dosed through the 20 milligram per kilogram PMO equivalent dose in the MAD portion of DELIVER. Most treatment emergent adverse events were mild to moderate in severity, with one serious treatment emergent adverse event that was judged unrelated to study drug by the study investigator, and this was dehydration due to gastroenteritis. There were no reports of anemia, thrombocytopenia, kidney injury, and very importantly, there were no clinically meaningful changes in electrolytes such as magnesium. The safety profile has supported dose escalation through that 20 milligram per kilogram PMO equivalent dose, and approximately 275 doses have been administered in DELIVER as of the data cutoff date. Next slide, please. Turning to dystrophin expression as measured by Western blot, which is also the co-primary endpoint of DELIVER along with safety and tolerability. Now, the gray bar represents patients in the placebo-treated group in DELIVER, and the blue bar represents the Dyne 251 treated patients in DELIVER. As you can see here, patients treated with Dyne 251 had a mean absolute dystrophin level of 0.88% of normal and a 0.28% change from baseline at 6 months, while the placebo group was largely unchanged. Now, on the right side of the slide, you can see data which was published in the Journal of Neuromuscular Disorders, where eteplirsen showed a mean absolute level of 0.3% of normal and a 0.06% change from baseline at 6 months. In looking at these two separate studies side by side, Dyne 251 appears to have a 2.5-fold higher dystrophin expression at 6 months than eteplirsen, with a 24-fold lower total PMO dose administered 4 times less frequently. Now, our goal for this initial data readout was to be at least as good as standard of care at 6 months with a very different dosing paradigm, which is once a month for Dyne 251. We believe that's meaningful for patients. So these data are compelling, and we believe we're on track to achieve higher levels of dystrophin in our dose optimization cohorts, which is the 10, 20, and 40 milligram per kilogram PMO equivalent dose cohorts, where we hope to see 5 to 10% dystrophin production at 6 months. We know from work of others in the field that dystrophin builds over time, and others have seen a doubling of effect out to 96 weeks. So we believe a 10 to 20% dystrophin production would be significant to be able to alter the trajectory of the disease for patients. Next slide, please. So next, we looked at percent dystrophin positive fibers, or PDPF. This measure is really important because quantifying localization of dystrophin to the muscle cell membrane helps provide confidence that there should be functional benefit. Again, the gray and the blue bars represent placebo and the Dyne 251 treated groups respectively in DELIVER. And you can see that Dyne 251 demonstrated 22.2% mean level of dystrophin positive fibers and a 19.8% change from baseline at 6 months. In the eteplirsen trial, again, published in the Journal of Neuromuscular Disorders, eteplirsen showed a 19.6% mean level of PDPF and a 10.7% change from baseline at 6 months. Next slide, please. So to summarize these compelling DELIVER data, what we showed is that at Dyne 251 dosed monthly at our initial 5 milligram per kilogram cohort showed an increase in percent dystrophin at 6 months. Similarly, this dose level showed an increase in percent dystrophin positive fibers at 6 months. And we've also demonstrated a favorable safety profile as of the data cutoff date, and our 20 mg per kg cohort is currently fully enrolled. So what's coming up in 2024? Well, in 2024, one of our key goals for the DELIVER study is to optimize the dose and dose regimen. So we will continue in this study to get to that optimal dose and dose regimen. And then we will also come back to this community in the second half of 2024 with another update in terms of our biomarker data, safety tolerability, and other data. Next slide, please. We believe this data set from DELIVER illustrate the potential of the FORCE platform in developing next generation exon skipping therapies and continuing to build on Dyne 251 to address other exon mutations. Historically, muscle delivery has been the challenge, and these initial data of Dyne 251 demonstrated targeted delivery to muscle with the potential for a wide therapeutic index where other approaches have struggled. And Dyne is committed to developing a DMD portfolio leading with Dyne 251, and we've identified PMOs for mutations amenable to exon skipping, skipping exons 53, 45, and 44. Next slide, please. And finally, we have tried to take every opportunity to listen to the Duchenne community on their needs for next generation treatments. We are working urgently to deliver for the community, including families and healthcare providers, to bring a potentially meaningful treatment forward. And I have to thank the families for being generous with your thoughts, advice, and for the sacrifices you continue to make to advance the science and advance medicines. And on behalf of Dyne, I would like to thank you all for your time and attention to listen to this presentation. Thank you again to CureDuchenne for having us here. And Catherine and I are happy to take any questions. Thank you.
Thank you so much, Ash, and welcome, Catherine. Um, Before we jump into the Q&A, I just want to remind everyone that to enter your questions, you need to do so via the Q&A button at the bottom of the screen, and you can be entering them as we're talking. So we had some questions come in beforehand, and so I think
Great.
I'll just get started on some of those. And maybe we could just start with, in general, how is your modality in terms of muscle delivery different than some of the other exon skipping approaches in the clinic?
Sure, sure. Happy to take that. That's probably one of the most common questions we get. And we can spend a whole hour sort of dissecting the various modalities, but I'll try to be succinct here. So as I mentioned, we have three components: the Fab, the linker, and then the antisense oligonucleotide. The FORCE platform is modular. So for all our therapeutics, we use the same Fab and the same linker for our investigational medicines, and we change the antisense oligo depending on the genetic basis of the disease that we're trying to target. So in terms of what differentiates us from different modalities, there are some companies that, for example, use a full monoclonal antibody to target the transferrin receptor. And we use that top portion that I mentioned. So what we believe the advantage is here is a smaller overall investigational medicine, right? 2/3 the size, or 1/3 the size, right? In terms of the monoclonal antibody. It also doesn't have the FC domain. So we also believe there should be a much lower potential for immunogenicity and effector cell activation. Again, these are things that we'll continue to see as we generate more clinical data. But so far, we're able to see a pretty favorable safety profile as of the data cutoff date. We also use a clinically validated linker. So this is a linker that is used in products that are currently on the market in the US for oncology, antibody drug conjugates that are used in oncology. And so there are other modalities that use a novel linker, for example, and that just adds a little bit to what needs to be further investigated as opposed to using something that's clinically validated. And so I think the other piece of the FORCE platform, of course, is the use of the transferrin receptor. Again, we're using the body's natural cell biology. The transferrin receptor, the TFR1 receptor's function is to essentially maintain iron homeostasis. It's used for iron metabolism. And so this receptor exists on all of these muscles that are key in terms of the pathology of Duchenne. And so we use this natural mechanism to bring that PMO into the cell. It forms that endosome, and then without any membrane destabilizing agents, it just exits the endosome and travels to the nucleus. So we don't need to have other attachments that will create pores or holes in the cells to allow the PMO to get into the muscle cells. And what often happens is it gets into other organs as well, and it can lead to unintended side effects. So again, our FORCE platform is designed to use the body's natural cell biology and not lead to unwanted off-target effects. And so we'll continue to update the community on our safety profile, but we feel good about what we're seeing, and we continue to dose escalate.
Thank you. And just to pick up on one thing that you said, that this transferrin receptor is on all the muscle groups that we sort of want to target. So we're talking about skeletal muscle, we're talking about heart muscle as well, diaphragm. So
Skeletal muscle, including diaphragm, cardiac muscle. And one thing that we don't talk a lot about, I think, is also smooth muscle as well. And so we are also able to address, because of the use of the transferrin receptor, potentially GI issues or other issues as well, because we do also get into the smooth muscle too.
No, that's wonderful. And I think, certainly across a number of neuromuscular diseases, right? This would have a big impact.
Yeah,
so,
like myotonic dystrophy type 1 and others, absolutely.
exactly.
And I can refer the community back to preclinical data that we have generated and communicated that has looked at cardiac muscle and diaphragm specifically. Obviously, we are not doing biopsies that allow us to specifically test what's happening in the diaphragm and cardiac muscle in our first in human study. We do have measures of function. Maybe what I will say is we are collecting those outcomes though. So we are looking at forced vital capacity percent predicted, other measures of pulmonary function, echocardiogram, cardiac MRI. So we are in our DELIVER study looking at clinical functional outcomes in terms of cardiac function as well as pulmonary function.
Well, since you mentioned that, there's another question here asking about whether you're also looking, are you doing MRI imaging to look at muscle in a more global way than just aside from localized
Yeah,
biopsy?
it's a great question. So in DELIVER, we are not doing muscle MRI. Again, we're doing cardiac MRI. We have a number of measures for muscle strength and function. Many that this community is familiar with, like NSAA and the time function test. But also we're using like the side device to look at stride velocity and more of a continuous measure of function, if you will. So we are trying to be thoughtful about what kinds of measures we're looking at in DELIVER, and certainly we're thinking about how we might incorporate muscle MRI in the future.
Terrific. Maybe we can talk a little bit about the clinical trial design. So you're currently in the sort of dose escalation phase of it. And there's a question here, and if you can't answer it, I'm sure you'll tell me, are you already enrolled at the higher doses, or is that coming? Can you speak to where you're at?
Yeah, sure. Yeah, we have enrolled the higher doses. As I mentioned, so the answer is yes. As I mentioned, the 20 milligram per kilogram cohort is fully enrolled, and we'll be back to this community in the second half of this year with more data. So right now, I can't say much more beyond that, but enrollment has been going on very well, and we look forward to communicating more data in the second half of this year.
And you can't say any more about what kind of data that we would maybe see later this year?
So we haven't publicly committed to data beyond safety, tolerability, and dystrophin. But as we get closer to that time, we may release a little more information about exactly what kind of data that we'll be releasing.
Okay. And there's some questions here, just to be clear. When you switch to the sort of open label or the long-term extension,
Yeah.
is that still placebo controlled, or does everyone at that point receive drug?
Yeah. So my apologies if I wasn't clear. So once an individual is completed the 24-week placebo-controlled portion, they then enter the open label extension, and everybody receives active treatment. So everybody receives Dyne 251 once they've completed the 6-month or 24-week placebo-controlled period. I will also say that in the lower dose cohorts, we have a 2-to-1 randomization. So meaning, for every 2 patients on Dyne 251, we have a patient on placebo. And at the higher dose cohorts, it becomes a 3-to-1 randomization where we have 3 patients on Dyne 251 and 1 on placebo. So it's also not 1-to-1, 251 and placebo.
Mm-hmm. Thank you. And is there the consideration of potentially during that long-term extension, moving individuals up to higher doses if it seems that it seems safe and more effective?
Yeah. Yeah, no, a great question. The answer is yes. So built within the protocol is to move patients up to the next higher safe dose. So we have that option in the protocol, yes.
Thank you. I think that addresses a lot of what has come in. I'm just going to scan through a little bit here.
Maybe while you're looking, Catherine, is there anything from these questions that you want to add to or modify?
No, I think you've done a great job, Ash. I think there's some questions, Liana, that you're looking at as well, kind of looking, I think this is from one of our colleagues at PepGen around manufacturing challenges based on the complexity of the technology.
Sure. So I think it's a great question. I will say that our CMC group is incredible. To that person's point, we are combining sort of three components for our investigational medicine, which is no small feat. And we are in a great place to supply our clinical trials as we've planned and even think beyond that. So currently there's no manufacturing challenges that we've encountered that are of note, and we're excited about the ability to serve patients through our trials and beyond.
Terrific. There's also a question here, I think just clarifying in terms of whether the trial enrollment is full or closed, or is it still actively recruiting?
Yeah,
I didn't want to switch over to clinicaltrials.gov, so maybe you
it's
can
still
answer.
actively recruiting, yes. So if you go to our corporate website, you can actually see a slide that really details the stepwise approach in terms of the dose cohort and the dose escalation. I apologize. I wasn't sure about time, so I didn't include that in this deck here, but it is on our corporate website. So the 20 mg per kg cohort is fully enrolled, but we're continuing to enroll in the trial. Yep.
Terrific. And question here from one of our pulmonary clinicians, I think we all know well, and I think you mentioned, so is pulmonary function, maybe you could speak a little to what's being tracked throughout the study.
Sure. So as has been done in many other trials, we are looking at forced vital capacity percent predicted, as well as FEV. So we are collecting a few different measures of pulmonary function. And I think maybe provide some context around this. So as I mentioned, we have a broad age range. One of the things that frankly, Liana, you at CureDuchenne, others have taught me is we need to be able to serve a broader set of the Duchenne community, especially the transitional and non-ambulatory group. And so Dyne very purposefully started this trial with that fairly wide age range of 4 to 16 years of age. And because there are some measures that are more applicable depending on what stage you are at in the disease. So for example, if someone is non-ambulant, perhaps doing an NSAA doesn't make any sense, isn't going to, help you understand kind of the treatment interventions effect. So we have pulmonary function in there, and it's extremely important because that pulmonary function is relevant, across the age range essentially. And there is literature, for example, that supports between ages 10 and 18, regardless of ambulatory status, there's essentially a linear decline of FVC percent predicted of 5 to 6% annually. So can one attenuate that or even sort of stop that decline with an effective treatment intervention? So it's really important for us, especially with this broad group, to look at measures like pulmonary function to be able to understand what's happening almost to some extent, regardless of the ambulatory status.
No, that's terrific. And just to say, thank you to Dyne for listening to the community and actually, enrolling such a wide range of ages. And, it'll be exciting to see that data when it's ready. So the other question we often get, and you said you already have some programs to tackle other exon skipping amenable populations. What about being able to skip more than one exon at a time?
Yeah, it's a great question. And I think I've gotten this a lot, and I think I clearly, I haven't provided a good enough answer. So for Dyne 251 specifically, we're looking at deletion mutations that for which an exon 51 skipping treatment would be appropriate. So our treatment is not exploring individuals who might need a double skip. Now, there is a little bit of data out there from other not other programs necessarily, but there's academic literature out there on double skips. There's academic literature out there on duplications, intronic mutations that act as deletions, for example. So there is some information out there. On the double skip, I think there are probably people that are much more appropriate than me to talk about how that could be addressed, such as Professor Anamika Artsmarus, Dr. Steve Wilton. I think, they are definitely knowledgeable in this space and can help help folks understand what it takes for double skipping. Now, from my basic, sort of perspective, there is a requirement of, the kinetics of, being able to have that double skip with two different antisense oligonucleotides. And how do those kinetics work? Do they work in tandem so that you can actually, skip have the skipping that you want without having no effect or some detrimental effect? And so there's a lot of work that needs to be done, I think, preclinically, maybe before we even get into the clinic in terms of double skip. But there are definitely some others that have far more expertise in this than I do.
Well, I appreciate the thoughtful answer because I think we're all looking at the literature on that, but to know that you guys are actually considering all the pros and cons and what would be the challenges you need to be able to solve to get there.
I mean, for us, we're just really trying to make sure that with Dyne 251, the platform is doing what it needs to do, which we believe it is so far. We're getting into the muscle. We are able to have robust exon skipping, generate dystrophin at our lowest dose, the 5 mg per kg dose, and also show benefits in terms of like dystrophin positive fibers as well. So it's really important for us to continue to build the body of evidence around Dyne 251 and to think about how that applies to other exons and then other sort of maybe other things like double skip.
Thanks. And just because the question was coming in, I'll let you reiterate again the other exons that you're looking, the programs you already have going, and anything about the timeline.
Sure. So I had mentioned in the presentation, having identified payloads for 53 skipping, 45 skipping, 44 skipping. I don't have specific timelines that I can offer, at this time. Again, hopefully in the latter part of the year, we can provide some more information. But I will also say that we presented in vitro data or early data for our 53 and 45 program at World Muscle a couple of years ago. So we're continuing to, move those programs forward.
Terrific. And then this last question is a little philosophical, I think, but I'd love to hear, I think everyone's
I'm going to
asking
ask Catherine to answer it
question.
anyways, so it's okay.
So it's a question of how, Dyne 251 compares to, gene therapy. And, a lot of individuals are thinking about this, having conversations with their physicians and, thinking about how effective, how durable. So, I know what I said, it's a little bit of a philosophical question, but I'd love to hear your thoughts.
Sure. Let me provide some thoughts. I'm going to ask Catherine to also participate here because I know she has a very strong perspective and like me, a lot of experience in this space on the gene therapy side as well. So in terms of just the modality, just so we're level setting, as we've talked about, this is an antisense oligonucleotide designed to skip, designed to address exon 51 skip amenable mutations. So it's very specific to that. We are also looking to produce a shortened functional form of dystrophin. Now, with gene therapy, as I'm sure most of you know, it is a viral capsid that has a portion of the gene, a transgene, with other elements tied to the transgene to make sure it gets to where it needs to go to and it turns on appropriately, so that a microdystrophin or a mini dystrophin can be produced. And there is that data out there that shows both the benefits of that as well as some significant safety issues that have arisen with gene therapy. And please know, we hope all of these modalities continue to progress and make it to market, from not just from Dyne's perspective, but I'm sure from everybody in industry. We want more therapies, more options for families. So I think that there are very, very big differences between those modalities. So this viral capsid elicits an immune response naturally in individuals who get it, so they can't be, at least at this time, be redosed, for example. To your point, Leanna, exactly what you said, it does appear that there are still significant questions around durability of efficacy, and is it waning over time, as some of the boys who were treated some years ago are now out, 4, 5, 6 years, and we're able to see a little bit more in terms of durability. And so it is not possible to redose right now. So the question is, what else can they be administered to be able to, continue to have a good progression, or to be able to stem the progression of disease. And so those studies, those experiments, I feel like they need to be done. People need to understand, like in the SMA space, is there a role for combination therapy? Is there a role for both antisense oligonucleotides, as well as gene therapy? You see in the SMA space, there's small molecules, antisense, and gene therapy, and they're used in combination. So what does that treatment paradigm look like? And so that data just needs to be generated, whether in a controlled environment, or in terms of just the real world evidence that will be generated over time. Ours is a chronic therapy, right? Ours is a chronic therapy designed to, to generate that more sort of native form of dystrophin, if you will. And so far, at least with Dyne 251, we're not seeing sort of immunogenic effects and the like.
So can I
Catherine?
just build on that, Ash? Because I think, at the root of the question is really, how do we make decisions for ourselves and our families around treatment choices or participation in research, right? And for those of us who like to make data-driven decisions, when we think about our own risk-benefit calculus, um, it's really, to what Ash said, getting that data out in the public domain so that people can make those decisions, right? I think it's very early days for gene therapy, so there are uncertainties, but we should be encouraging that data be in the public domain as soon as possible. I think we've had some therapies on the market, right, where we could certainly kind of learn from that experience if some of the outcomes were made available, right? So that folks are today in a position to make better decisions around their health and for their children.
Thank you for that.
That's
I know
very
it was a very loaded, very complicated question that I asked you, but I think your thoughts are very much appreciated for both of you.
Right, and it's
And
platforms like this that ultimately are going to be able to disseminate that information, right, to the patient community as they are struggling with these decisions before them.
absolutely. And, I think so. A couple more questions have come in, if you don't mind, since we got some time still. And I think the answer might be, we'll see from the data, but there's a question here about with the skipping platform, whether or not it works the same way or is as effective if you're just trying to skip, if you have only one exon deletion, or if you have a multiple exon deletion and then skipping. And the example this question came in was saying, say like you have a deletion from 45 to 52,
right.
and you're skipping 53. And so maybe you could speak a little bit to the context of that in terms
Sure.
of what you're doing when you're skipping, and what do we know and not know.
Yeah, it's a good question. Definitely complicated question. So I guess maybe where I would start with this is, there is literature out there that suggests different levels of functionality in terms of dystrophin isoform, or the type of dystrophin that's created, depending on what is skipped. So it is not necessarily true that if it's a deletion of exon 52 versus 45 to 52, right, that dystrophin that's produced by skipping will be the same. So there is some interesting literature out there for 51 skip equivalents, 53 skip equivalents that help one sort of understand potentially some of those differences. So I think that is in terms of what functional benefit that confers. That said, what you can see pretty reliably is the dystrophin production. So whether or not you have a single deletion or multi-deletion for a particular type of mutation, you're able to produce with the right kind of intervention and measure the dystrophin restoration in that muscle tissue. So that is why dystrophin is still considered, as I mentioned earlier, by KOLs, an absolutely key outcome of what needs to be looked at in terms of treatments for Duchenne. And so that is also why we are looking at dystrophin production in Dyne 251 across a spectrum of ages, status of the disease, and various deletion mutations as well. So it's a really good question because I think there's probably, the literature suggests that you may have different functionality with different types of dystrophin created from skipping, depending on the deletion mutations that one has. I think the other piece of this that's not asked, but I think is important is there is also literature out there that helps one understand with different dystrophin levels at baseline what one's phenotype may look like. So for example, if you have 0% or 0 to 0.5% or 0.5 to 1% or over 5, there is data out there. It's cross-sectional in nature, so not longitudinal. But it's cross-sectional that shows that at this band there is a longer time to loss of ambulation. It confers benefit in terms of survival. So it's also clear, not surprisingly, that the more dystrophin one has, the more benefit it has, from a functional point of view. And even low levels of dystrophin can confer some benefit over time.
No, that's terrific. And I think we will all be anxiously or excitingly awaiting the data towards the end of second half of this year, and and continuing to look across the whole Duchenne community, right, of data coming in from different modalities. It's an exciting time, I think, to see how this all plays out.
It is exciting, complicated. We're here to answer any questions. I think Leanna, Catherine, and I will be in front of this community whenever you want us to be in front of the community and an update on our program.
Well, I want to thank you both again today for making time and for answering so many of our questions. I think it's been terrific, and I just how much we appreciate you coming and speaking to our community directly. And for all of those of you watching on your computer, we also thank you again for making time in your day to share this with us. In terms of logistics, there's going to be a post-webinar email that'll be sent tomorrow in case you have any other questions that come up that you didn't have a chance to ask, and we will pass those along. We will also get in touch to let all the attendees know where they can see the webinar once it's posted online, so that way you can review it again or share it with anybody else that you think might be interested. And with that, I just want to say thank you to everyone, and have a good day.